Okay, hello everyone, welcome back. So let's continue here. Just make sure I am sharing the correct screen. Okay, so I believe we left off when we were about to start talking about working distance. We just talked about magnification as well as field of view. So now we're going to talk about working distance. So working distance is the distance between the specimen and the objective lens. So if you have your specimen here and your objective lens is over here, this would be referred to as your working distance. As you can see, when you are using your 4x scanning objective lens, you have a greater working distance between the lens and your sample. As you increase in magnification, this working distance is going to decrease. The high power objective lens has to be closer to the specimen than the low power in order to focus. So we know as magnification increases, our field of view decreases, and now as magnification increases, our working distance also is going to decrease. So this is really important when you're using a microscope to be just aware of your working distance, because when you are using your 4x objective lens, you can use your coarse adjustment knob to move your stage up and down because you have a greater working distance. When you are using your 10x or 40x objective lens, you are not to use your coarse adjustment knob because that moves the stage quite rapidly and you run the risk of crashing your stage or your specimen against the objective lens. So you're going to damage both the objective lenses as well as your sample. So that's why when we are using the 10x or 40x objective lens, we only use our fine adjustment knob. The fine adjustment knob will move much slower than our coarse adjustment knob. Okay, so for activity 4, you guys are going to follow this link and use a virtual microscope. If the link does not work, just copy and paste this into your browser. You will need Adobe Flash in order to use the microscope, and you will want to click here where it says Start Tour to familiarize yourself with this microscope as well as all the functions of this microscope. So very briefly, you will need to turn your microscope on. This will allow the light to come from your microscope through the diaphragm. You will want to adjust your lighting. I usually start off somewhere in the low to medium range. And then you adjust your diaphragm, your aperture setting, so you determine how much light you want to let in. You will always put your microscope at 4x. So make sure your microscope is at 4x. Bring your stage all the way down so you have a huge working distance here. Clip in your microscope slide using these clips. The video will show you how to properly clip in a microscope slide. And then you're going to want to look at the eyepieces and make sure you adjust those to your face. So when you look at the eyepieces and you see two circles, like a circle here and a circle here, you will need to adjust those and bring those in closer until you get one circle. So once that is adjusted to properly fit your face, you are going to want to, you should already have it at 4x, but make sure your microscope is using the 4x objective lens, and you will use the coarse adjustment knob to bring your stage all the way up. You also want to center your specimen, so you want to make sure that the center part of your specimen is right below your objective lens. And you will center it using the X and Y axis to move this platform stage. So now we're going to have our platform stage all the way up, and we're going to slowly move our coarse adjustment knob down. So that's going to bring the stage down away from our lens. So as you bring it down, whenever you see your slide in view, you're going to stop and you're just going to use the fine adjustment knob to fine focus it. Once you reach what you think is the best, the most resolution, you will use this revolving nose piece to go to your 10x objective lens. And there you should just need to use your fine adjustment knob to focus your image. You might have to change the lighting. Usually as you go up in magnification, you need more lighting to come through. So you might move the condenser or just make it brighter down here. So then once you feel like your image is focused at 10x, you're going to move up to 40x. Here, once again, you're only going to use your fine adjustment knob and focus your image. If you use a microscope this way, you will always be able to find your image. You should have no problems. So once again, you always start with your 4x. You start with the stage all the way up. You bring your stage down slowly until you find your image using the coarse adjustment knob. And then you move to your fine adjustment knob and fine focus it. You move from 4x to 10x. You only use your fine adjustment knob here to focus. To 40x, only use your fine adjustment knob here to focus. So here we're just going to go over a little bit of the compound light microscope. So you want to plug the microscope into the electrical outlet and flip the switch on to turn on the light source. The light source shines from the bottom of the scope up to the stage. The stage is a platform where microscope slides are placed for viewing, so you will place your microscope slide here on this platform stage. The light travels towards the stage from the iris diaphragm. It has a small lever that allows you to regulate the amount of light traveling through your specimen. So there will be a lever on this diaphragm so you can control how much light is going to pass through to your slide. This is very similar to using a camera. So in photography, you have aperture setting where you can control how much light you want to come in. The image passes through the objective lens attached to the revolving nose piece. So this here is your revolving nose piece. These are the objective lenses that are connected to your revolving nose piece. The image can be brought to sharper focus using the coarse adjustment knob and then the fine adjustment. So always begin with coarse, then go to fine, always begin at your 4x microscopes, objective lens, I'm sorry. The image passes through the ocular lens, which has a magnification of 10x. So your oculars will have a magnification of 10x. And the microscopes in Bio 1 have two ocular lenses. They are binocular scopes. So as you can see, you have two eyepieces. Some microscopes have one eyepiece. I've actually never used a microscope with one eyepiece, but I have seen them. And it is important to make sure that the lenses stay clean. If you must clean the lens, you have to use the specific lens paper or else you run the risk of scratching your lenses. So focusing the light microscope, the compound microscope has three objective lenses. You have your 4x scanning lens. This magnifies your specimen 4 times. Your 10x is called your low power lens. This magnifies your specimen 10 times. The 40x is a high power lens. And remember to never use the coarse adjustment at this magnification. I don't even use the coarse adjustment at 10x. I really never have a need for it. I can do everything with just the fine adjustment knob. So I never use it. Always begin with the 4x lens. If the object is out of focus, use the coarse adjustment to bring the object into focus. The coarse adjustment knob moves the stage closer or further from your objective lens. You can only use the fine adjustment at 40x. So this just remember, at 40x, only use your fine adjustment. I actually only use fine adjustment at both 10 and 40x. Once your specimen is in focus at 4x, you can move up to the 10x objective lens. Always be aware of your working distance space. Again, use the fine adjustment to bring the specimen into focus. You may find that a higher magnification needs more light. Finally, to see the most detail, use the 40x objective lens. Only use the fine adjustment at this setting. So this kind of will talk you through how to use a compound microscope. So keep this in mind when you are doing your activity. So the first thing you want to do is switch on the power and adjust your lighting to a medium setting or so. You want to make sure your microscope is set on the 4x scanning lens. You will put your microscope slide on the stage. You open the clip and slide your sample to the back of the stage. Make sure it does not go under the clips. The tutorial should show you how to do that properly. You will begin using your 4x objective lens. You will use the course adjustment to bring your stage to the highest position. And you typically want, sorry, I have large dogs, so they're pretty loud. So you will use your 4x objective lens. You use the course adjustment to bring your stage all the way to the top. You typically want your condenser light to its lower point here. You don't need too much light when you are using a low magnification. You center your sample under the lens using your XY axis knobs. And then you will use the course adjustment knob to bring your stage down slowly. When you're looking through the eyepieces and you notice, I see my specimen, then you will just use your fine adjustment knob to fine tune your focus. Once you're happy with the image you have at 4x, you will move up to your 10x objective lens. You will move the condenser accordingly depending on how much lighting you need. And you will use your fine adjustment knob to focus your specimen here. Then once you're satisfied at your 10x objective lens, you move up to your 40x objective lens. You put the condenser at the highest position, so typically you need more light here. And then only use the fine adjustment knob here to bring your image into focus. So when you guys are when you guys go to that site, you guys will be able to look at a microscope slide that has this E on it. So put this microscope slide into your microscope and complete the 2 questions and drawings on page 18. You will also watch this YouTube video. I'm not too sure if I play it here, if you're going to hear the audio, but I'll post this YouTube video up, and this is basically what we would have done if we were on class. You would use a toothpick to swab inside your mouth, and you would be collecting human cheek epithelial cells. You would put that on a microscope slide and then stain it with a specific dye so that we can visualize our cells. This is called creating a wet mount, and you will put a cover slip on top of it and clean up any mess if needed. The stain you would be adding is methylene blue. And you would just use the microscope to identify the nucleus, cytoplasm, and other cell structures of your epithelial cells. And then you would draw the image in your lab notebook. I believe it's page 20. But watch this video to see how you would prepare your slide. And then using that same link, you will follow this link and you can select the cheek smear slide to see what it would look like under a microscope if we were in lab. And then you will draw the cheek cells at 40x in your notebook. Remember just to use the Start Tour button if needed. And if you guys have any questions or run into any problems using these microscopes, let me know. I followed these links and used the microscope prior to making these videos, and I found it pretty straightforward and easy to use. So hopefully it's easy to use on your computer as well. But if you guys have any questions, let me know. And that is the end of our presentation. Don't forget your guys' lab worksheet 2. Lab worksheet number 2 is due Friday, September 4th, as well as your quiz. Both of those will be up on our main bio page. The supplemental information or the lectures, the videos, all of that is under our lab group page. Hopefully you guys all are familiar now with how we're doing the Canvas site and you guys understand where to find the material. I do recommend doing the lab worksheet prior to doing your quiz. It will give you a general understanding of what questions will be on your quiz. Some things on your quiz are from the slide or from the presentations I post, and they're not in your lab manual, so you guys are required to watch these presentations. And let me know if you guys run into any problems or have any questions, okay?